Our next speaker is Andrew Krutchinsky, and he will be telling us about his efforts to interface the MultiQ IT with the Orbitrap.
Thank you, Liz. I want to continue to talk about a new type of ion trap that we've been developing in Chait lab of Rockefeller University. I will be talking about tandem multi-quadrupole ion trap. I will tell you why do we develop this ion trap, on what principle it operates, and how it can be useful for mass spectrometry. Let me start with the HPLC blank. Here you can see a total ion current of a chromatogram that we ran almost 2 hours. No sample was ejected. At any moment of time, full MS spectrum would exhibit quite abundant peaks that you're probably familiar with, and they represent clusters of probably liquid and impurities in liquid and in a gas phase. And at any moment of time, whether 10 minutes or 60 minutes or 110 minutes, the spectrum would be sort of the same, and it's quite intensive. If you look down in a grass, we will see that lots of other small peaks. And if we imagine that we have a peptide there, it will be easy to miss it because of so many chemical noise species. We reproduce the situation by taking a sample and diluting it to the point where we stop seeing a signal of 5 peptides. For example, this is a region where we should be able to see 5 peptides, but we don't see. Instead, we see the grass of singly charged ions, and that spectrum was obtained in the mass spectrometer that I will be talking today about. And yet, we can convert this spectrum with the push of 2 buttons into this spectrum, and where all 5 peptides are easily detected, and the signal to noise is improved by more than 200 times. We achieved that by building a new type of ion trap, but we call it multi-quadrupole ion trap. And the major idea we build in this trap is to enable massively parallel mass spectrometry. Let me tell you how it's built. So we take a multitude of cylindrical electrodes and put it around the volume where we meant to hold the ions. For example, this construction would take 488 electrodes, and you can see that construction without front panel removed. We drive it for now with sine waves, 2 sine waves shifted by 180 degrees. And one can show by computer simulations that this structure would stably trap ions for a long time. For example, this is a trajectory of single ion with parameters indicated here. That was trapped for more than half a second. And we could continue further on. If we play with the parameters here, and we might notice that if we reduce potentials from, say, from 150 volts to 50 volts, interesting things happen. The ions that will thermalize quickly and then will migrate along the volume slowly. It will occupy bigger volume. And eventually, it might find a space between the quadrupoles suitable for it to visit, because it will have stable trajectory there. And if it continue moving, it may exit the device. And that creates multitude of inputs and outputs for ions to enter and exit the device, and actually produces the basis for parallelization. This ion trap has several modes of operation. Let me illustrate that. When ions enter the ion trap, say with the energy of few electron volts, they largely neglect the exits of the ion trap. But when they thermalize, they might start visiting the spaces between, and then will hit the supporting plate, for example. To prevent that, we put a small potential difference between a set of quadrupoles and the supporting plate. To provide ions out, we make a hole and use extended quadrupole, and we call it trivial MOSE mode. That's how ions enter and exit the device. What we want, however, we want to put several quadrupoles, and set these small quadrupoles such that to transmit specific ions in a specific MOZ ranges. That will enable us to split the ion beam that enters the MultiQ ion trap into subbeams containing ions from non-overlapping MOZ regions and create a system of concurrent beams that can be analyzed simultaneously. I'll be talking this later. So, much easier mode to organize and observe is the Z mode. All what we needed to observe this mode is to reduce this potential from 2 volts to a few hundred millivolts. What we think happens is we create a small bump inside of the quadrupole in the order of kT, where K is the Boltzmann constant and T is temperature. Ions that thermalize to kT, those ions that have kT will pass this barrier, and those are singly charged ions. They have energy kT, the height of the barrier is kT, they can pass it. But not multiply charged ions, because they will see the height of the barrier as proportional to their charge. For example, triply charged ions will see a height of the barrier as 3 kT. So that produces effect that lets us to get rid of singly charged ions preferentially and keep inside multiply charged species. Remember, we have 486 exits, and that enables us to multiply effect to the point where we observe it as ions enter and exit the device. So I have sometimes compare this to online dialysis system. It's equivalent. Interestingly, MultiQ ion trap, this type of we think that this type of ion trap have similarity to a system that all of us have in a huge number of copies, billions, we believe. And this is a cell nucleus. Cell nucleus also can compartmentalize different compounds, either temporarily or permanently. For example, nucleolus and DNA stays in the cell nucleus. But many molecules are shuttling in and out of the cell nucleus through the nuclear pores, which approximately in each eukaryotic cells, approximately from a few hundred to 1,000. And you could imagine that this shuttling has to be fast enough to provide for the need for cell to act properly. Mechanism of gating of these molecules at nuclear pores was proposed about 16 years ago by Brian Chait and co-workers, and it has entropical nature. In case of MultiQ ion trap, the gating is electrical in nature and manifests itself in a Z mode or MOZ mode. Interestingly, that elements of theory can be found in this book, Random Works in Biology by Howard Berg. What's more even important that we can find some formulas there that can easily be checked experimentally, and that's what we're going to do. Let me show the results. So we built a tandem MultiQ ion trap, an electrospray interface, and put it in front of LTQ Orbitrap XL. So ions produced by electrospray source introduced through the heated capillary into quadrupole ion guide. Then they go through the skimmer, go in the first MultiQ ion trap, reside there for some time. Then they transfer freely, no pulsing or nothing. They go to second MultiQ ion trap, and then they proceed to spectrometry where they are analyzed either in linear trap or Orbitrap. And let me remind you why we wanted to build a tandem configuration. And we had two, at least, purposes. One is we wanted to, previously I showed that single MultiQ ion trap lets us to get rid of singly charged ions by a factor of 30 to 50. And we thought that if we put two together, we will get a very high depletion efficiency because the two processes are independent, we will have multiplicative of depletion efficiencies of each device. And second idea is we want to provide parallel channels between two MultiQ ion traps and use first MultiQ ion trap as an ion beam splitter and the second MultiQ ion trap as an ion beam combiner to assess the performance of parallel channels. Here's just a part of the spectrum that contains at least 2 peptides, lysine and cephalin, and frequently another peptide that manifests itself in the charged state 4. We're frequently interested in this small guy. So we can convert this spectrum into that spectrum. And, sorry. And a relative ratio of 4+ ions increased 1,000-fold. And that's just done with the push of 2 buttons. This is how experiment is performed. We run, for now, infusion, direct infusion, 5 peptides mixture. That's this concentration 100 femtomole, for example, direct infusion with 500 nanoliter per minute. First, the ions pass through MultiQ IT trap unaffected. This is this part. Then we switch, say, first instrument in depletion mode, in Z mode or depletion mode. I freely substitute that 2 terms. Then we switch second MultiQ IT trap in depletion mode, and then return back. Here's just spectrum, single scan, just to emulate sort of fast acquisition and during HPLC run. It's a good spectrum. All peptides are there, but we also observe few abundant singly charged species. When we go to first depletion mode, the signal of lysine and cephalin, not signal, but the flux of ions of lysine and cephalin drops by almost 40 times. And if we go to the second, we switch to multi-Qs in depletion mode, we practically get rid of all singly charged species. And you can see that how much we enriched, not enriched, you see clearly multiply charged species. This mode of operation, in this case, flux was reduced by 300 times, and for charged species increased by three times, which is, I cannot explain right now. We cannot explain right now. But most importantly, this type of depletion of Z mode is very useful when we analyze very small amount of samples. We diluted the sample 100-fold, and you can see that we stop seeing peptides, or mostly singly charged species. And when we set 2 multi-Q ion traps in depletion mode, we got all spectra, all peptides back, and plus more. Our postdoc, very talented postdoc, Junjie Wang, he produced several computer programs that allow us to analyze spectra in the presence of singly charged to produce statistical analysis of the spectra, to see how many singly charged, multiply charged ions we see. So we run these programs on this type of spectra, and we showed this and other experiments. And we showed that we can see 10 times less +1 peaks and 2 to 10 times more multiply charged peaks, usually more highly charged peaks. Now he's putting an HPLC system in front of this instrument where we hope to analyze, and actually he did already analyze in his initial experiments, where he would use very small amount of sample and use depletion mode to observe this sample. So in conclusion, and the future work, so we started with Z mode because it's a low-hanging fruit. It's easy to produce for us. And indeed, we tested the hypothesis, previously stated hypothesis, whether depletion coefficient of both devices would be multiplicative. And it looks like it's true. We do see the effects of charge space that approximately start when 10⁻⁹ ions per second passing through the first multi-Q ion trap. But we have several ideas how to overcome that and go even probably higher in the charge states or number of charges, to handle number of charges. One possibility is to introduce multiple parallel channel between 2 devices. That should allow us to offset the space charge effects. If we provide more outputs from the first device, we will reduce the number of ions at a given time in the first device proportionally to the number of parallel channels we introduce. Also, most importantly, these parallel channels will allow us to finally to evaluate MOVZ mode, where our major goal is to split ion beam into sub-beams containing ions from non-overlapping MOVZ regions to produce concurrent beams that can be analyzed simultaneously. Or that means that enabling parallel mass spectrometry. We think that parallel mass spectrometry can be the basis for the next generation mass spectrometry tools. By analogy, that next generation DNA sequencing. Because these tools should enable us to comprehensively analyze single cells. We think that there are three elements important for this, to reach this goal. First, a beam needs to be transferred into mass spectrometer with 100%, and we have some ideas using conduct electrodes, for example, how to do that. I just showed you that it's possible to use MultiQ technology to filter almost 100% of background ion species. But most importantly, this technology should be able to enable us to split ion beam into multiple sub-beams, which can be analyzed by other mass spectrometers, maybe sequential ones. But it can be shown that if the number of parallel channels is 1,000, then all sample components can be analyzed simultaneously with the fraction of time needed to analyze just one. Let me just show you several examples of what next generation mass spectrometry tools based on an idea of parallelization would look like. For example, based on multi-Q ion trap, if we use ions, we let the ions in multi-Q ion trap, then we split it on, say, 10 beams. We set them into 10 collisional cells and analyze it either 10 quadrupoles or 10 linear traps, boosting efficiency at least 10 times and sample usage, because everything is done simultaneously. It would be better to analyze it in the 10 orbit traps or single orthogonal injection multi-beam time-of-flight mass spectrometer. I'd like to thank the organizers of the conference for giving me this opportunity to talk today, and Liz, and thank you for your attention.